Now let's move on to another very important topic for the ATP paper, and that is food tests. This is one of the most commonly tested topics and is usually worth several marks, so you should know it very well. Before carrying out any food test, remember the sample preparation. If the sample is solid, crush it using a mortar and pestle until it forms a fine paste. If the sample is liquid, dissolve it in distilled water if necessary. Always use a small amount of the sample to obtain accurate results. Now let's look at the important food tests you need to remember. For the starch test, use iodine solution. If starch is present, the sample turns blue-black. If starch is absent, it remains yellow-brown. For the protein test, use the biuret reagent, which contains sodium hydroxide and copper sulfate. A purple color indicates the presence of protein, while a blue color shows that protein is absent. For the reducing sugar test, use Benedict's reagent. Heat the mixture in a hot water bath, never over a direct flame. A positive result may appear green, yellow, orange, or brick red, depending on the amount of reducing sugar present. If no reducing sugar is present, the solution remains blue. For the fat test, also called the emulsion test, add ethanol to the sample and shake it well. Then pour the mixture into cold water. A cloudy white emulsion indicates the presence of fats, while a clear solution indicates that fats are absent. If you would like a more detailed explanation of these food tests, you can refer back to Chapter 4, Biomolecules, where each test is explained thoroughly. There are also a few additional tests that are important for the ATP paper because the exam may ask about their procedure, observations, or expected results. The leaf starch test begins by boiling the leaf to stop all chemical reactions. Next, place the leaf in hot ethanol to remove the chlorophyll. Then rinse it in hot water to soften the leaf before placing it on a white tile. Finally, add iodine solution to test for starch. You should also remember the carbon dioxide tests. The hydrogen carbonate indicator turns purple when the level of carbon dioxide decreases and yellow when the level of carbon dioxide increases. If you need to keep the carbon dioxide level constant during an experiment, use sodium hydrogen carbonate or potassium hydrogen carbonate. If you need to remove carbon dioxide, use sodium hydroxide or potassium hydroxide. Another traditional test uses lime water, which turns milky when carbon dioxide is present.